Introduction. Welcome, medical students, to today's lecture on the intricate world of biochemistry. In this detailed script, we will explore the fundamental aspects of DNA structure and organization, gene expression regulation, transcription, RNA processing, the genetic code, ribosomes and translation, tRNA and aminoacylation, initiation and elongation of translation, protein folding and modification, chaperones and quality control, degradation of proteins, and finally, we will conclude our discussion. Let's dive right in. DNA, or deoxyribonucleic acid, is a remarkable molecule that carries the genetic information necessary for the development and functioning of all living organisms. It consists of two long strands that form a double helix structure, with nucleotide building blocks comprising a sugar phosphate backbone and nitrogenous bases. The four nitrogenous bases, adenine, thymine, cytosine, and guanine, pair in a complementary fashion. Adenine links with thymine, and cytosine pairs with guanine, forming hydrogen bonds. This precise base pairing allows for DNA replication and faithful transmission of genetic information. Furthermore, DNA is packaged into chromosomes, which are highly organized structures found within the nucleus of eukaryotic cells. These chromosomes undergo intricate folding and coiling, facilitated by DNA-binding proteins called histones. This organization ensures that DNA is efficiently compacted, safeguarded, and readily accessible during gene expression processes. Gene expression regulation is a fundamental process that controls when and to what extent genes are transcribed and translated into proteins. It allows cells to respond to environmental cues, maintain homeostasis, and undergo differentiation. Several mechanisms govern gene expression, including transcription factors, DNA methylation, histone modifications, and non-coding RNAs. Transcription factors are proteins that interact with specific DNA sequences, termed enhancers or promoters, either activating or repressing gene transcription. DNA methylation involves attaching a methyl group to the DNA molecule, resulting in gene silencing. Histone modifications, such as acetylation or methylation, can alter chromatin structure and gene accessibility. Transcription is the process by which DNA is used as a template to synthesize RNA molecules. It involves three main steps: initiation, elongation, and termination. During initiation, RNA polymerase recognizes and binds to a specific DNA sequence called the promoter, marking the starting point for transcription. Elongation encompasses the actual synthesis of the RNA molecule, with RNA polymerase moving along the DNA template and synthesizing RNA in the 5-3 direction. Finally, termination occurs when specific sequences or proteins signal the end of transcription. After transcription, the newly synthesized RNA molecule undergoes a series of modifications collectively known as RNA processing. These modifications include the addition of a 5-cap, splicing out introns, and adding a poly-A tail to the 3-end. The 5-cap and poly-A tail play crucial roles in mRNA stability and transport, while splicing removes non-coding regions, introns, and joins the coding segments, exons, together. This process allows for increased protein diversity by alternative splicing, where different exons can be included or excluded from the final mRNA. The genetic code is a set of rules that determines how the nucleotide sequence of mRNA is translated into the amino acid sequence of a protein. Codons, composed of 3 nucleotides, serve as the words of this code. There are 64 possible codons, which encode the 20 amino acids found in proteins, along with 3 stop codons that signal the termination of translation. This redundancy allows multiple codons to specify the same amino acid, providing some protection against mutations. Translation is the process of protein synthesis and occurs on ribosomes, large molecular complexes composed of RNA and proteins. Ribosomes consist of 2 subunits that assemble around the mRNA molecule. It involves 3 main steps: initiation, elongation, and termination. During initiation, the small ribosomal subunit, along with several initiation factors, recognizes the start codon on the mRNA. Then, the large ribosomal subunit joins, and translation begins. Elongation involves the stepwise addition of amino acids to the growing polypeptide chain. Finally, termination occurs when a stop codon is encountered, releasing the newly synthesized protein. Transfer RNAs, tRNAs, are small RNA molecules that carry specific amino acids to the ribosome during translation. Each tRNA has an anticodon, a 3-nucleotide sequence that base pairs with the corresponding mRNA codon. The attachment of the correct amino acid to the tRNA is catalyzed by aminoacyl tRNA synthetases, ensuring accurate pairing between amino acids and codons. This process is known as aminoacylation. The initiation of translation involves the assembly of ribosomes on mRNA and the selection of the correct start codon. In prokaryotes, the small ribosomal subunit binds directly to the Shine-Delgarno sequence located upstream of the start codon. In contrast, eukaryotes use a different mechanism involving the binding of several initiation factors to the 5-cap structure, followed by scanning of the mRNA until the start codon is identified. During elongation, amino acids are added one by one to the growing polypeptide chain. Each amino acid is delivered by a specific tRNA, guided by the complementary pairing of the anticodon with the mRNA codon. The process involves elongation factors and GTP hydrolysis. Termination occurs when a stop codon is encountered, and it is recognized by release factors that promote the release of the completed polypeptide chain from the ribosome. After translation, the newly synthesized polypeptide undergoes folding into its functional three-dimensional structure. This process, known as protein folding, is essential for proper protein function. Molecular chaperones assist in this process, preventing incorrect folding or aggregation. Additionally, proteins may undergo post-translational modifications, such as phosphorylation, glycosylation, or lipidation, to acquire their functional diversity. Chaperones are a class of molecules that aid in protein folding and stability, ensuring correct protein conformation. They assist in the folding of newly synthesized proteins and help in the refolding of misfolded proteins. Additionally, chaperones play a crucial role in quality control by recognizing and targeting misfolded or damaged proteins for degradation. Protein degradation is a vital process that maintains cellular homeostasis. The two major pathways involved are the ubiquitin proteasome system and lysosomal degradation. The ubiquitin proteasome system involves the tagging of proteins with ubiquitin, which serves as a signal for degradation by the proteasome. Lysosomal degradation, on the other hand, employs enzymes within lysosomes to break down proteins into their constituent amino acids for recycling. In conclusion, biochemistry provides insights into the intricate mechanisms that govern fundamental processes in living organisms. From the structure and organization of DNA to the synthesis, folding, and degradation of proteins, these processes are tightly regulated and interconnected. Understanding these principles is essential for unraveling disease mechanisms, developing targeted therapies, and advancing medical research. I hope this detailed script has helped you gain a deeper appreciation for the complexity and importance of biochemistry in the field of medicine.